Hello, my name is Sydney Ghazarian, and in this presentation I'll be discussing my capstone project titled Post-Escort Recruitment by STIV, Effects of Site-Directed Mutagenesis on the Major Capsid Protein. I know the title is a lot to unpack, but I hope to be able to make it more clear with this presentation. In my lab, I study viruses, specifically a virus that infects archaea. But what are archaea? Archaea are one of the three categories all living things can be classified as. They are a small microorganism that looks similar to bacteria, but their molecular processes are more closely related to eukarya. Archaea tend to be classified as extremophiles, as they can be found in deep-sea hydrothermal vents and acidic hot springs. However, they can be found in non-extreme environments, such as soil. For my study, I used the archaeal species Sophilobus sophotaricus. The species was originally isolated from the acidic hot springs in Yellowstone National Park and can be found in environments that are acidic and high in temperature. Sophilobus is infected by a virus known as STIV, or Sophilobus turreted icosahedra virus. It gets its name from the small projections, or turrets, found on its surface. To understand the architecture of the virus, shown in blue is the major capsid protein encoded by the gene B345. In yellow is the host-derived internalized cell membrane. And in the middle is where the double-stranded DNA of this virus is found. STIV is considered a model archaeal virus, however, its replication cycle has not been fully characterized. My lab aims to determine the mechanism behind each step in this cycle. Specifically, my project focuses on the packaging and assembly of STIV. During an STIV infection, it has been found that there is an upregulation of the host escort3 gene cluster, indicating that STIV may be utilizing escorts during its replication cycle. Escorts, or endosomal sorting complexes required for transport, are proteins that are used in many cellular apposition processes. One example is during cell division, where these proteins act as the final pinchers to separate the cells into two. Interestingly enough, some viruses can even hijack these escort proteins to continue infecting other cells. Escort proteins are found in all eukaryotic cells and in some archaea. Eukaryotes have 4 different escort proteins, while archaea have only been found to have 1, which is a homologue of escort3. This suggests an evolutionary relationship between archaea and eukarya that can be explored to further reveal the divergence of the two domains. Previous studies have shown that STIV's major capsid protein, B345, interacts with escort3. We hypothesize that STIV's major capsid protein helps to recruit escort3 proteins to vesicles that will eventually become the internal membrane of the assembled virion. This interaction was abolished when the C-terminus of B345 was removed. The goal of my study is to determine which amino acids in the C-terminus of the major capsid protein are necessary for its interaction with escort3. For the study, I could have created mutations in the B345 gene of STIV's genome. Although this is not impossible, it is difficult to achieve due to the large genome. Instead, I inserted the B345 gene into an archaeal expression vector, PSESD1, and performed PCR mutagenesis with the primers that I designed. From there, I transformed the vector into E. coli to achieve a higher concentration of the vector. I then re-isolate the vector and send it for sequencing to verify that my mutation occurred. After going through these steps and troubleshooting along the way, I was unfortunately unable to make these mutations in the B345 gene. If my lab had not been shut down due to the COVID-19 concerns, I would have instead attempted to use CRISPR to create the intended mutation. From there, I would transform the mutated vector into Sophilobus and induce the production of B345 using arabinose. From there, I would perform a Western blot to determine if the major capsid protein was being produced, and run a pull-down assay to determine if escort3 B345 interactions are maintained. I just want to take a moment to thank everyone in Dr. Jamie Snyder's lab for all of the support and mentorship that I received during this process. I would have not been able to continue my project without them. I would also like to thank the MBRS RISE program with providing me the funding to perform this research. And finally, I would like to thank the Honors College for giving me the opportunity to perform my research project on campus. Thank you for listening.